Hi everyone, my name is Adrian and today I'll be covering figures 3A through E. And so before I give the overview of this section, I'd first like to touch upon what was discussed in the previous section. So the researchers saw that impairment of IL-33 signaling led to significant decreases in the T cell count, the T cell proliferation, and T cell production of interferon gamma. And so after this, they wanted to investigate to see if impairment of IL-33 had other effects on other cells. So specifically, they looked at the myeloid lineage cells during brain T-gon-di infection. And so the authors noted that macrophages are important to control T-gon-di proliferation because they secrete a molecule called iNOS. And so this will be covered more in detail with my group member Kevin in the next few slides after me. What I will be covering is how the researchers investigated the role of IL-33 signaling in recruiting monocytes from outside of the brain in order to combat T-gon-di infection. And so the way they did this was they used two different experiments, sorry. They used flow cytometry and a CCR2 RFP reporter mouse in order to investigate potential differences in infiltrating monocyte recruitment between the wild type and the IL-1 RL1 knockout mice. And so this is the first experiment that I'll be discussing, and this is the second experiment with the CCR2 RFP reporter mice. Okay, and so I'll just give a quick breakdown of the flow cytometry markers that were used in order to more easily understand the results of figure 3A and 3B. And so one of the key issues in the flow cytometry experiment was to distinguish microglia from infiltrating monocytes. So microglia are the brain resident macrophages, so they are already found in the brain, whereas infiltrating monocytes are found outside the brain. And so both of these cells are myeloid lineage cells, and they both express CD11B, which are the blue trapezoids here. And so this marker is enough to distinguish them from lymphocytes, but it's not enough to distinguish them between each other. And so to distinguish them, they used a different marker called CD45. And so CD45 is an activation marker. And so it is actually expressed more uh, it's actually expressed higher in infiltrating monocytes compared to microglia. So here we have more CD45 expression compared to microglia, which only have two. And so because of this, microglia are called CD45 intermediate, whereas infiltrating monocytes are called CD45 high. And so with that, we can go on to figures 3A and 3B. And so what we have here is the results of multiple brain samples from each group. So the first group is the wild type mice, and the second group is the IL-1 RL1 knockout mice. And so on the y-axis, we have CD11B, which is the marker for the myeloid lineage cells. And on the x-axis, we have CD45, which is that activation marker. And so we can see here on the top right, they designated this group of cells that had this signal as infiltrating myeloid cells. And this was because they have high CD45 expression, and they also have high or positive CD11B expression. And so these cells were called infiltrating myeloid cells based on the high expression of both markers. To the left of them, we have the microglial cells, which have slightly lower CD45 signal. And so because they still have the CD11B signal, these are the microglial cells that have intermediate expression of CD45. On the bottom, we have lymphocytes, which aren't too important for what I'm covering here, so I'll just ignore these two. And so what the researchers did was compare the signals between the wild type and the IL-1 RL1 knockout mice. And if you look at the numbers here, 26%, 4%, you can see that these are the amount of cells, these are the percentage of cells from the total that are in this category. So 26% of the total cells are infiltrating myeloid cells, and they are within this gate. and 4% of the total cells are microglia and are in this state. And so we can see a decrease in infiltrating myeloid cells with a drop of 17% versus 26%. And that is actually seen a bit more easily in panel B here. And so on the y-axis, we have the cell number, and on the bottom here, we have the different markers. And so for the CD45 positive cells, we see a significant decrease in the IL-1 RL-1 knockout mice compared to the wild-type mice here in black. And so when they looked at the CD45 intermediate cells, or the CD45 intermediate CD11b plus cells, which are the microglial cells here, they did not see any significant differences between the two groups. Um, and then when they saw, or when they investigated CD45 high cells, CD11b plus, they noted again a significant decrease in the IL-1 RL-1 knockout mice versus the wild type mice. And so what this suggests is that by knocking out or impairing the IL-33 signaling in these mice, they somehow have an impaired recruitment of monocytes to the brain in order to fight T. gondii infection. One other quick thing to note is that this CD45 plus significant difference is probably driven in part by the lymphocytes. And so I'm not too sure what to make of this, although I think that's what is causing that significant difference. I think it is more important to note this results here with the CD45, CD11b plus cells, since these are the infiltrating myeloid cells that we are interested in, and which are the cells that could potentially fight off T. gondii or help in the fight against T. gondii in the brain. And so what they did next was to look at this notion or to explore this idea in a different way, just to further validate or lend support to this notion that IL-33 signaling mediates or helps to recruit infiltrating monocytes from outside to the brain. And so they used a CCR2 RFP reporter mouse. And so these mice have CCR2, which is their receptor for CCL2, that important ligand that we learned about in the last paper. So they have this receptor fused to a red fluorescent protein. And so when you image it by fluorescent microscopy, you get a fluorescent signal. And so another important thing about this reporter mouse is that CCR2 is expressed only on infiltrating monocytes and not on resident microglia. So essentially, wherever you see CCR2 RFP signal, that'll come from a monocyte and not from a resident macrophage microglia. And so what they did is that they took multiple brain sections of CCR2 RFP wild type and CCR2 RFP knockout mice, and they imaged these brain sections by fluorescent microscopy to visualize and quantify the differences in monocyte recruitment. And so here's a very crude schematic of what I was just talking about. All right, and so here are the results from 3C and 3D. So on the top, we have the wild type mice, and on the bottom, we have the IL-1 RL-1 knockout mice. And so right from the beginning, we can see a clear difference between the two. In the wild type mice, you have lots of CCR2 RFP signal, and specifically in this box, which is zoomed into on the right, we can see plenty of the CCR2 RFP signaling. In contrast, on the bottom, in the knockout mice, we already see a very significant stark contrast versus the one on top, versus the wild type mice. And if we look at the zoomed in box here to the right, which is the same area, we already see far less CCR2 RFP signaling as well. And so this is, again, can be quantified. And on the y-axis, we have the volume coverage. And so basically, this is the amount of CCR2 RFP signaling, but just in number form. And so what we see again is a significant difference in the IL-1 RL-1 knockout mice compared to the wild type mice. And so this again adds to the notion or to the idea that The IL-1 RL-1 knockout mice, since they have impaired IL-33 signaling, they also have impaired recruitment of infiltrating monocytes that could potentially fight off the T-Gondii infection. And so, just to give a quick summary of what I just mentioned, the researchers performed two experiments, flow cytometry and fluorescent microscopy, to see how impairment of IL-33 signaling in the IL-1 RL-1 knockout mice, how that impairment would affect their ability to recruit monocytes to the brain in order to fight off the T-Gondii brain infection. And so, although they demonstrated a reduced monocyte recruitment in these mice, in the knockout mice, they would also want to investigate if the immunological phenotype or processes of these cells is also affected by impaired IL-33 signaling. And so they did this in the next set of experiments by looking at INOS production from macrophages, and that will be covered by my group member Kevin after me. So thank you very much for listening and have a wonderful day. Thank you.